Hello and welcome to Symbiotic class. Friends, today we shall discuss about symbiotic nitrogen fixation. Before that, I request all of my friends, those who have not subscribed my YouTube channel, please subscribe. Symbiotic nitrogen fixation is the part of a mutualistic relationship between certain microorganisms and higher plants in which the microorganisms dwell within specialized structures such as root nodules, pre-existing stem glands and others as symbionts, and fix atmospheric nitrogen into ammonia, which is directly provided to the host plant in exchange for nutrients. The most common type of symbiosis occurs between the members of the plant family Fabaceae, also called Leguminosae, and soil bacteria of the genus Mesorhizobium, Azorhizobium, Bradyrhizobium, Cynorhizobium, and Rhizobium, which are collectively called Rhizobia. Another type of symbiosis occurs between several woody plant species and soil bacteria of the genus Frankia. Still other types of nitrogen fixing symbiosis involves South American herb Gunnera and tiny waterfall Azolla, which form associations with Cyanobacteria nostoc, which form associations with Cyanobacteria nostoc and Anabina. Finally, some nitrogen fixing prokaryotes are also associated with C4 grasses such as sugarcane and miscanthus miscanthus. Symbiotic nitrogen fixation is the part of a mutualistic relationship in which plants provide a niche and fixed carbon to the nitrogen fixers in exchange for fixed nitrogen. Symbiotic nitrogen fixing bacteria invert the roots of plants and induce the formation of specialized structures called nodules, which contain nitrogen fixing bacteria. Nitrogenase is the enzyme which fixes atmospheric nitrogen. It contains specialized sites that facilitates the high energy exchange of electrons. Oxygen, being a strong electron acceptor, can damage the sites and irreversibly inactivate nitrogenase. Hence, nitrogen fixation must occur under anaerobic conditions. Symbiotic nitrogen fixation occurs within specialized structures. Some symbiotic nitrogen fixing bacteria dwell within nodules, which are specialized structures that develop on roots of plants when infected by nitrogen fixing bacteria. In Gunnera, nitrogen fixation occurs within pre-existing stem glands which develop independently of the symbionts. In case of legumes and actinorhizal plants, the nitrogen fixing bacteria induce the development of root nodules. Grasses also form symbiotic association with nitrogen fixing prokaryotes, but in such cases root nodules are not formed. Nodules contain oxygen binding heme proteins called leg hemoglobins. Leg hemoglobins are the most abundant proteins in the nodules, crucial for nitrogen fixation. It has a high affinity for oxygen. Its function is to increase the rate of transport of oxygen to the respiring symbiotic bacterial cells, which decrease the steady-state level of oxygen in infected cells. To continue aerobic respiration under such conditions, the bacteroid uses a specialized electron transport chain in which the terminal oxidase has an affinity for oxygen which is even greater than that of leg hemoglobin. The symbiosis between legumes and rhizobia is not obligatory. Legume seedlings germinate without any association with rhizobia and may also remain unassociated throughout their life cycle. Rhizobia also occur as free-living organisms in the soil. Under nitrogen-limited conditions, however, the symbionts seek other through an elaborate exchange of signals. Signaling, the subsequent infection process, and the development of nitrogen-fixing nodules involve specific genes both in the host and the symbiont. Formation of root nodules. Two processes. One is infection and the other one is nodule organogenesis occur simultaneously during the development of root nodules. Rhizobia usually infect root hairs by fast releasing node factors which induce a pronounced curling of root hair. Rhizobia become enclosed in a small compartment formed by curling. Also, in response to node factors, the cell wall of the root hair in this region is degraded, allowing the bacterial cells a direct access to the outer surface of the plasma membrane of the root hair cells. The next step is the formation of infection thread. The infection thread grows at its tip by the fusion of Golgi-derived membrane vesicle at the end of the tube. Deep within the cortex near the xylem, the cortical cells dedifferentiate and start dividing, forming a distinct area within the cortex called nodule primordium, which gives rise to root nodules. Different compounds acting positively or negatively control the development of nodule primordium. Node factors activate localized cytokinin signaling in the cortex and pericycle of the root, leading to the localized suppression of polar oxygen transport, which in turn induce nodule morphogenesis and stimulate cell division. The infection thread filled with proliferating rhizobia elongates through the root hair and cortical cell layer towards the nodule primordium. When the thread reaches the nodule primordium, its tip fuses with the cell membrane of host cell and penetrates into its cytoplasm. Bacterial cells are subsequently released into the cytoplasm surrounded by cell membrane of host cell. This results in the formation of an organelle called symbiosome. Branching of the infection thread within the nodule enable bacteria to infect many cells. At first, bacteria within symbiosome continue to divide and the symbiosome membrane increase in surface area to accommodate the growth by fusion with smaller vesicles. Soon thereafter, because of an undetermined signal from plant, These bacteria stop dividing and differentiate into nitrogen fixing bacteroids. Nodules as a whole also develop vascular system for exchange of nitrogen fixed by rhizobia for the nutrients produced by plants. It also develops a layer of cell to exclude molecular oxygen from the root nodule interior. Both non-symbiotic and symbiotic nitrogen fixation is catalyzed by a metalloenzyme known as the nitrogenase complex. It is oxygen sensitive and catalyzes the ATP-dependent reduction of molecular nitrogen into ammonia. Reduction of molecular nitrogen into ammonia, a 6-electron transfer, is coupled to the reduction of 2 protons to evolve S2. The overall reaction catalyzed by the enzyme nitrogenase complex is N2 plus 8 electron and 8 protons plus 16 ATP gives rise to 2NH3 plus H2 and 16 ADP plus 16 inorganic phosphate. The enzyme nitrogenase complex can be separated into 2 components. One component is the Fe protein and the other component is MOFE and the other component is MOFE protein. In the overall nitrogen reduction reaction, pyridoxine serves as electron donor to Fe protein, which in turn hydrolyzes ATP and reduces MOFE protein. Binding of ATP to Fe protein and its hydrolysis to ADP and inorganic phosphate is thought to cause a conformational change in the Fe protein which facilitates the redox reaction. So Fe protein reduces MOFE protein, which then can reduce numerous substrates. But under natural condition, MOFE protein reacts only with N2 and H+, as a result of which molecular nitrogen is reduced to ammonia. So friends, this is all about symbiotic nitrogen fixation. For more updates, please subscribe my YouTube channel, Symbiotic.